Yeah, so my name is Marcelo. Thank you for having me. And the title of my presentation is Comprehensive Investigation of Pesticides in Honey, Pollen, Bees, and Soil Collected from Canola Fields in Saskatchewan. So before I start my presentation, I would like to introduce myself and our researcher group. So I'm new here in Canada. I'm from Brazil. And my research in honeybees started in 2016 at São Paulo at São Paulo State University, where I did my master's and my PhD. And then last year, on July, I moved to Canada to start my postdoc here at University of Saskatchewan. They didn't tell me about the winter, so I arrived in July. And here, it's our group. I'm glad to be part of this amazing team. And this team is led by Dr. Sarah Wood and Elimir Cinco. And if you want to know more about us, you can use this QR code to check our website. So back to my presentation. First, I will start talking about pesticides and honeybee health, and my research purpose and plan, and how we can use pollen collected by honeybees to characterize the floral resource, and at the end, wild pollinators surveillance. So canola is made in Canada, and not surprising, Canada is the largest producer and exporter of this crop, and there is a sweet and lucrative relationship between honeybees and canola. So pollination services here in canola can increase the yield estimated in $1.7 billion per year. And we can also use the honeybees to produce hybrid canola seeds, which is estimated in $1.4 billion every year. On the other hand, for beekeepers, 70% of honey extracted here in Western Canada comes from canola. So wherever you find canola, you will find honeybees. Here in the Western Canada, on the prairies, the great majority of canola is grown from seed treated with neonicotinoids insecticides to protect this crop from insect pests like flea beetle, but it can also be a potential risk to pollinators because in majority they are also insects. And there is so much controversy regarding this neonicotinoids regulation. In 2013, the European Union limited the use of neonicotinoids, and the canola producers experienced some economic adversities due to the high losses caused by pests. Here in Canada, in 2018, the Health Canada proposed to discontinue the use of these neonicotinoids, the thiamectoxam and clothianidin, and they are very important for the protection against the flea beetle. And then after 3 years, they decided not to implement any change for use of neonicotinoids in the canola fields. So it can change again. And one of the important questions here is how we can maintain this equilibrium between the crop protection against flea beetle and the preservation of pollinators' health and biodiversity. So after 5 years of study here, Dr. Sarah Wood from University of Saskatchewan reported that honeybees exposed to 5 to 10 nanograms per grams of neonicotinoids did not experience negative effects. But these colonies exposed to 20 nanograms per grams of neonicotinoids experienced negative effects like decreased honey harvest, reduced population, and increase in overwinter mortality. So these results leads us to think about what's the level of pesticides our colonies are exposed here in Saskatchewan. So there is only one study here in central Saskatchewan published in 2016 that reported some alarming levels of pesticides in 30 samples of honey and 20 samples of pollen. And when we compare these results to the average worldwide here, we can see that 60% of samples are more than that average, and even higher than 20 nanograms per grams of neonicotinoids. So if these colonies are exposed to this high level of neonicotinoids, they should experience these negative effects. But in contrast, what we see here in canola fields are colonies growing up fast and producing lots of honey. So maybe, due to the small sample size of this research and limited geographic area, maybe this is not representative for all the province. Another fact was the same year of this research in 2016, the average honey produced per colony here in Saskatchewan was 200 pounds, the highest average in all the country. So, due our observations, we have hypothesized that the total pesticides level on canola fields in Saskatchewan is comparable to the rest of the world and below these 20 nanograms per grams of neonicotinoids. So, the objects of this study, the main goal is to perform the largest study in Canada to document residues of 215 pesticides in samples of honey, pollen, bees, and soil. The second goal is to characterize the floral research and non-agricultural vegetation using pollen collected by honeybees and the aerial data sets. And the third goal of this project is to make a wild pollinator surveillance and compare the diversity of these wild pollinators with the pesticide levels we found and the plant's abundance and diversity in each area of study. And to make that possible, in collaboration with 15 canola growers, we will have 15 sample sites here in the Saskatchewan canola belt from Lloydminster to Yorkton, and another 5 sample sites in the Boreal region that will be our control group, so at least 10 kilometers from any kind of crops. And so we start this research on this summer and the next year. For that, we will use a total of 80 beehives, 4 per each site, and we will collect a total of 2,208 samples of pollen, honey, bees, and soil at 3 different times, before, during, and after the canola bloom. So all the samples will be sent for the pesticide residues analysis. So the honey will be collected directly from honey frames, and then each hive will be set up in a hive scale for monitoring the colony weight gain and estimate the honey harvest. We will collect soil at a depth of 10 centimeters and using vacuum to collect the honey bees that returns from the field, and pollen traps to collect the pollen. So once we collect all the pollen, it comes mixed with every flower that the bees visited. And we want to know the level of pesticides we have only in canola and then in the rest of this pollen. So using this palynological analysis under the microscope, we can see the morphology of the pollen. So this is a pollen from canola, and we find another pollens that be collected, and we can identify using the morphology of pollen grain. So we have a survey of the floral research the bees are collecting from. And to confirm this analysis, this visual analysis, 10% of all the samples, of all the samples here will be subjected to a DNA metabarcoding analysis. So basically we can extract the DNA from the pollen, and then with sequencing method we identify this plant species and compare to the palynological analysis to see how reliable it is. Then, once we have identified all the floral research, we will use 2 data sets, drone to take some aerial pictures in each field, and then QGIS software to create some diversity vegetation maps to compare for each region and boreal forests. Then, for the third goal, we will use some pun traps, 10 pun traps per site to collect these insects, so they are attracted for these traps, then fall into these bowls, and we can pin them and identify, then sort by pollinators and non-pollinators to compare this diversity with the pesticides levels we found and the plant abundance and diversity in each area. So with this research, this study will contribute to evidence-based pesticide regulation, which will enhance the current agricultural and beekeeping practices. So thank you again for having me, and if you invite me next year, I can show some results. Oh, please.
You're just looking for any pesticide residue there?
Yes, will be like 250 residues analysis. We can see everything, and we also are considering all the hybrids, so we want to see the real levels of everything here in Saskatchewan. Oh.
Do you expect to see any neonicotinoids?
Yeah, so because they use in seed treatment here, clothianidin probably.
That's a long time from seed treatment to flowering, though.
Yeah, that we'll see.
That one in the back there. Megan, go ahead.
So this comes in from one of the people in the webinar, and they're requesting that you show the honey hive province slide again.
Oh. Is the map here?
I'm going to say yes because they didn't say anything different. There's no question, just a request for the slide. So.
Oh, the beehive?
Um,
No, I don't know.
yeah, honey hive province slide.
Ah, sorry. Ah, the graph. Here.
Okay, thank you.
This-
They say perfect.
I'd suggest also going into the canola field with a sweet net while it's flowering, to make sure that the pollinators that are actually in the canola are showing up in your pan trap. Do you have that comparison?
Yeah, we are trying to collect in 3 times, before, during, and after canola. We'll do the same. Thank you. So,
Any more
Yeah.
questions? If not, then let's get out there early for lunch. And thank you very much.
oh, thank you.